Your body, even now as you rest, is not still. Beneath your skin, beneath the hush of muscle and the breath-soft rise of your chest, there is motion. Billions of cells, quietly working, repairing, remembering. They are not aware of you in the way you are aware of them, but they carry your instructions, your design-like notes folded gently into their pockets. Inside each one, a code sleeps, and you carry trillions of these tiny keepers. This code doesn't look like the alphabet you learned as a child, but it is made of letters. 4 of them, just 4. A, T, C, and G. Like the hum of a lullaby with only 4 notes, repeated in different rhythms, forming a song so long it could stretch across the stars if unraveled. These letters are not written on paper, but on a molecule called DNA, spiraling like a staircase within every cell. Imagine a ladder made of sugar and whispered in rails, curled into itself like a spiral galaxy. Each rung is a pair of those letters, matched always with their partner, with T. C with G embracing across the spiral like tiny hands. Your body is composed of trillions of cells, and nearly every one holds a complete copy of this code. Your personal instruction book, a vast, quiet library in miniature. In this code are the recipes for everything you are. For the curl of your hair, the way your lungs unfold, the exact shade of your skin beneath the moonlight. Each letter matters, each placement precise. Even a single change can ripple outward, like a pebble tossed into a still lake. As you breathe in now, cells in your lungs read from this ancient manuscript to build the proteins that make oxygen flow. As you rest, your heart cells follow the code that tells them how to beat, not too fast, not too slow. The language of life is read again and again, tirelessly, without complaint. You are built on repetition, on instruction, on a memory held by every cell of who you are, who you've always been. These letters came from somewhere, and that story is older than you, older than your parents, older than mammals, older than time measured by clocks. It is the story of life, passing itself forward, passing letters, one generation at a time. Sometimes faithfully, sometimes with errors, small typos, gentle shifts. These small changes, these mutations, are how evolution whispers. They are how giraffes grew necks long enough to kiss the tops of trees, how whales, once landwalkers, returned to the sea and remembered how to swim, how your eyes became able to read these words, generations after vision first learned to form. Your DNA is a time traveler, carrying messages from the deep past. It's a record of every ancestor that made it through the night, every creature that found a way to live, to eat, to breathe, to love, and to leave behind a child with the same song inside. It is no ordinary language. It does not rhyme or pause. It is 3 billion letters long, repeated in every cell, read by tiny machines called ribosomes. These machines never sleep. They do not complain, they do not forget. But the language, for all its brilliance, is not perfect. Sometimes mistakes happen. A letter is swapped or lost. Sometimes these mistakes are harmless, like misplacing a comma in a sentence that still makes sense. Other times, the result is more serious. A protein misreads, a process falters. A disease begins to form, quiet and slow. And that is why understanding these letters matters, because if we can read the story clearly, we can begin to understand where it goes wrong, and more gently still where it might be rewritten. You, lying here tonight, are made of countless chapters. Your liver tells one story, Your brain tells another. Your skin, your blood, your bones, they all read from the same library, but interpret it slightly differently, like actors playing from the same script with different roles. This is what makes you whole. This is what makes you you. And this is where CRISPR enters like an editor, arriving not with ink and paper, but with microscopic tools, ready to gently, precisely correct a single letter in a sentence that was nearly perfect, but not yet. Tonight, you are not an experiment. You are not a question or a project. You are a miracle of quiet design, alive in the stillness, with a code that sings softly in the dark. The cells do not know that you are thinking of them, but they are always thinking of you. Even as you sleep, they remember your shape. They remember how to hold you together. They rebuild what you lose. They fold and unfold instructions with grace, like monks in a temple copying sacred texts, tirelessly, lovingly. And it all begins with a T, C, and G. 4 letters, 4 choices, infinite combinations. Every sentence of your being born from their dance. Now, imagine, what if those letters could be changed on purpose? What if one day, a child born with a painful error in the script could be given a chance to read a different line, to walk through life without that weight? What would that take? Who would do it, and how? The answer begins not in a hospital or a lab, but in the strange microscopic world of bacteria, tiny ancient life forms who, long before us, learned how to fight back by remembering the invader's words and cutting them away. Let's follow them, gently, into that memory. Imagine a pair of scissors, impossibly small. They do not rest in drawers or belong to a desk. They drift in the microscopic ocean of a cell, invisible even to the most powerful lens. They do not cut paper. They do not know paper. But they are sharper than steel and more precise than a surgeon's blade. They cut molecules. They cut code, and they were not made by human hands. Long before science had a name for them, long before microscopes or molecules or medicine, these scissors lived quietly in the cells of some of Earth's tiniest creatures, bacteria. Not the bacteria that make you sick, but the kind that live in soil, in oceans, in your gut. The kind that quietly help the world turn. Even these small beings must defend themselves. In their world, danger often comes in the form of virus-sized tiny invaders that inject their own instructions into a cell, hijacking the machinery to make copies of themselves. A single infection can destroy a whole colony, but bacteria are clever. In their ancient struggle to survive, they built a memory away to recognize returning threats, like a notebook of viral faces tucked away in their genome. They called it, in the language of modern science, CRISPR, clustered regularly interspaced short palindromic repeats, a name that feels cold, like cracked ice, but its purpose is warm. With intention, CRISPR is not a thing, but a pattern, a structure, a vault of memories. When a virus attacks a bacterial cell, and the cell survives, it saves a tiny piece of the virus's code and tucks it into this archive. A mugshot in molecular form the next time that virus appears, the cell is ready. How? Enter the scissors. These aren't the kind you know. These are proteins enzymes with names like Cas9. They are designed to search, to match, to cut. Cas9 works with CRISPR like a guided weapon, but one that never explodes. Instead, it travels through the cell, carrying a small piece of RNA, a guide, a memory copied from the viral code stored in CRISPR's vault. Together, they search for a perfect match, When they find it, Cas9 unfolds its blades, and with a quiet molecular snip, the invader's code is broken. The virus can no longer replicate. The cell lives, and the memory remains. This, at its heart, is CRISPR's ancient brilliance. Not in its aggression, but in its remembrance, its ability to record danger and prepare for it. Not blindly, but specifically, one letter at a time. For centuries, this microscopic drama played out unnoticed, unseen. In the guts of bacteria and the waters of Earth, no one imagined that something so tiny, so old would one day become the centerpiece of one of humanity's greatest hopes. But science listens, and eventually it heard the whispers. In 2012, scientists Jennifer Doudna and Emmanuel Charpentier unlocked the secret of CRISPR's scissors. They saw not just a defense mechanism, but a potential tool. What if, instead of fighting viruses, these molecular scissors could be used to edit the genome of any living thing, not to hurt, but to heal? Suddenly, the world changed. CRISPR-Cas9 became more than a bacterial survival trick. It became a scalpel for DNA and a dream. Imagine being able to correct a single mistake in the genetic code of a human cell. A typo that causes a disease. A missing letter that leads to suffering. What if you could guide Cas9 to that one exact spot and fix it? You wouldn't need to rewrite the whole book. Just change one letter. And sometimes, that's all it takes to save a life. That is what makes CRISPR different from everything that came before. Older tools for genetic editing were clumsy, expensive, and imprecise, like using a chainsaw to trim a thread. CRISPR is different. It's specific, cheap, quick, and astonishingly accurate. But it's not perfect. Sometimes CRISPR cuts where it shouldn't. Sometimes the repair doesn't go as planned. And so, the journey is careful. Scientists are learning how to steer it better. To program the guide RNA, how to control the scissors, how to mend the cut in just the right way. Even now, as you rest, research continues. In quiet labs lit by blue light, in petri dishes and test tubes and mouse cells and human cells, dreams flicker through computers as lines of DNA are mapped and modified, and always, the hope is the same. To turn illness into health, to turn suffering into silence, to rewrite stories that once had only one ending. You might wonder, how do these scissors know where to cut? It's a question worth lingering on, because it leads us into the next chapter, into the memory banks of bacteria, into the guidebooks written in molecules, into the exquisite way that CRISPR finds its way through a sea of letters, to just the right line. But for now, let your thoughts settle. The scissors are quiet now, their hands folded, their blades resting in the shadows of possibility. Tomorrow, they will search again. But tonight, we listen for the guide. Before there were animals, before there were forests, before the sky turned blue with oxygen, there were bacteria, simple, small, nearly invisible, and yet they have outlived every age. They are the quiet architects of life, and tonight, you will follow them not with your feet, but with your thoughts. Bacteria are not the villains we often imagine. Yes, some cause sickness, but most are silent companions, helping you digest food, make vitamins, balance your skin. They have no brains, no hearts, no nerves, and yet they remember. Their memory is not emotional, it is molecular, a record, not of joy or pain, but of survival. Imagine a viressa, bacteriophage, a spindly-legged, alien-shaped speck latching onto a bacterial cell, injecting its code like poison ink, hijacking the machinery, turning the cell into a factory for copies of itself. For a bacterium, this is a death sentence, unless it remembers. This is where CRISPR begins, not as science, but as defense, as instinct, as an immune system without antibodies, as a notebook hidden inside the cell's own DNA. When a bacterium survives a viral attack, it slices a tiny piece of the invader's genetic code and stores it between repeating sequences in its own genome. Each piece becomes a page in a growing memory, a mugshot, a fingerprint, a fragment of threat. This archive is what scientists call CRISPR, not a tool yet, not a technology, just nature's ledger, a molecular scrapbook of battles fought and survived. And beside this memory sits a helper, Cas CRISPR-associated proteins. They are enzymes, workers, silent guardians that act when the alarm sounds, when a known virus returns. The bacterium copies the matching memory from its CRISPR bank. This memory becomes RNA, a flexible, one-strand note. It pairs with a Cas protein, forming a team. The RNA is the guide. The Cas is the cutter. Together, they search through every molecule, every twist of DNA, looking for a perfect match. When they find it, the Cas protein snips the viral code. Right there, right then, the virus is disarmed, and the cell survives. This is not revenge. It is protection, an instinct encoded in evolution itself. You may wonder, how can something so small, so primitive, be capable of such elegance? But elegance is everywhere in biology, especially in simplicity. Bacteria do not waste, they do not daydream, they build what they need and nothing more. The CRISPR system is modular, flexible, a kind of molecular memory drive with editable bookmarks. It is not one thing. It is a pattern, a network of functions spread across generations, and it works. That's why, across billions of years, bacteria have survived. Even as ice ages came and went, even as asteroids fell and continents shifted, even as all the great species rose and disappeared, they endured, editing their past into protection for their future. Now, scientists have begun to study these tiny engineers, not with fear, but with reverence, under microscopes, in petri dishes. They follow how bacteria remember, how they guide their Cas proteins like hounds, trained to sniff out danger. And in this ancient system, researchers saw something miraculous, a mechanism that could be borrowed, not just to protect bacteria, but to help humans. A mechanism that doesn't care what DNA it targets. As long as the guide matches, it cuts. This flexibility, this openness, is why CRISPR became the foundation for a new era of genome editing. All thanks to the bacteria, unseen, unthanked, but brilliant. Let yourself marvel at that, at how life's smallest forms hold some of its deepest wisdom. We often look to stars for wonder, but look inward. Look down to the soil, to the microbes in your gut, to the air you breathe. They are there, silent, old, and clever, and they do not just survive. They invent. CRISPR was never ours, it was theirs. We are merely guests in its story. Visitors, borrowing tools built by engineers we cannot see. But to use a tool well, you must understand how it works. That's what scientists set out to do, to understand CRISPR not as magic, but as a system, with components, with purpose, and the most delicate part of all, the guide. A strand of RNA that tells Cas where to go, what to touch, and where to cut. How does it find its way? How does it match a sequence in the vast sea of genetic letters? That is where we go next. But for now, rest in the idea that life has always been editing itself, and that the tiniest engineers have always known how. You are made of code. 3 billion letters curled inside every cell. They do not speak aloud. They do not shout. They hum softly, endlessly, like the quiet tune of your own biology. Amid this vast library, CRISPR must find one sentence. Not a page, not a chapter, one single line. Perhaps just a single word. How? How does a molecule the size of a whisper find its place in an ocean of text? The answer lies in pairing, in pattern, in memory. Let's return to the guide RNA, that single strand copied from the CRISPR memory bank. It holds a fragment of an old enemy, a viral sequence like a fingerprint pressed into clay. Attached to it is Cas9, the enzyme with the scissors. But the scissors are blind. They only know where to cut if they are led. The guide RNA is their map, but this is no map like the ones you've held in your hands. It doesn't show mountains or roads. It shows letters. A, T, C, G, over and over. 2. Find the target. The CRISPR complex drifts through the genome, like a feather floating through a book, landing, testing, moving on. At each place it stops. The guide RNA checks the letters, one by one. Are you the match? Do you remember me? If the letters don't align, it moves on. But if they do, if the match is exact, something delicate happens. Cas9 unfolds. It opens its blades and cuts, not in anger, not in haste, but with purpose, with focus, with a precision honed by billions of years of trial. To understand how precise this is, imagine this. You are standing in a library that holds every book ever written, in every language, every dialect, billions of pages, and you are asked to find a single sentence that begins with a single letter and ends with a sigh. CRISPR does this in moments. Why? Because it is built on the principle of base pairing. The same rule that lets a bind to T and C to G, the same dance that holds the double helix together. Base pairing is a lock and key, a left and right shoe. It works because the shapes only fit in one way. So when the guide RNA floats through the cell, it tests every strand of DNA for that fit. If even one letter is wrong, the fit fails. The blades stay sheathed, but if it fits perfectly, the site is opened, and the cut is made. This is why CRISPR is so powerful. Not just because it can cut, but because it knows where. Scientists can now design guide RNAs to target any gene in any organism with just a sequence, a string of letters. It's like programming a molecular search engine. You write the query and the system finds the match. But even with all this elegance, there are risks. Sometimes the match isn't perfect. Sometimes a similar sequence is close enough. The blades open when they shouldn't. A gene is damaged. A function lost. This is called an off-target effect. And so CRISPR is still learning, still being shaped by hands and minds who want it to be safer, cleaner, more faithful. Some labs are building new versions of Cas9, sharper, smarter, slower. Others are tweaking the guide RNA, adding new checkpoints, requiring more exact matches. Every improvement is a new verse in the poem of precision. You may wonder what happens after the cut. DNA doesn't like being broken. It rushes to repair. Like skin closing a wound, like cloth pulling its threads tight. Cells have repair systems. Some glue the cut ends together quick but messy. Others use a template to fix its lower but clean dot. This choice matters. Because if you want to edit the code, you can give the cell a new sentence, a corrected version. And if it uses that template during repair, the change becomes part of you. The code has been rewritten, a single letter changed, a disease prevented, a future restored, but it all begins with that search, that quiet molecular journey through a forest of letters, looking for one leaf. The guide RNA doesn't panic, it doesn't rush, it waits, it tests, it matches. This is the poetry of CRISPR, not in the cut, but in the finding. And perhaps there's something to learn from that. That healing begins not with force, but with recognition, with memory, with knowing where the pain is. Tonight, as you lie still, your own cells are doing this work. Reading, matching, pairing, guided by chemistry, but lit by something older. Every cell in you carries the ability to remember, to repair, to seek a better version of itself. And just as the guide RNA finds its way, so too does science. Now that it can cut with purpose, the question becomes, what should it change? The answers lie ahead in the soft space between suffering and hope, in the quiet decisions that begin when a body no longer works as it was meant to. But for now, let this idea rest gently in your mind. That precision can be soft, that searching can be sacred, that sometimes the smallest tool can find its way through the biggest maze, and tomorrow we will follow it into the body where the story of illness waits to be rewritten. Your body tells stories. Some you can see, like the shape of your smile or the rhythm of your breath. Others are written silently, deep in the folded strands of DNA, waiting to be read or misread. Sometimes a single letter in that code goes awry, not by intention, not by cruelty, but by chance, and with it, an entire story bends. You may never meet the child born with cystic fibrosis, but inside them, a tiny error keeps salt and water from flowing the way they should. Lungs thicken, mucus clogs, breath becomes labor. You may not know the teenager with sickle cell disease, but inside them, one small letter is swapped, and red blood cells twist into sickles, sharp and fragile. They cut from within. These are not rare myths. They are real. They are daily, and they begin with the same letters. We all carry a TCG, but in the wrong order at the wrong time. Now, science stands quietly beside these stories, holding a pen, not to erase, but to revise. CRISPR offers something medicine has long dreamed of, the chance to change the script itself, not to treat symptoms, but to fix the source, to find the misspelled word in a 3 billion letter sentence and correct it. In 2019, a young woman with sickle cell disease became one of the first to receive a CRISPR-based therapy. Doctors removed some of her bone marrow cells, edited their DNA in a lab, and returned them to her body. New authors with a better story to tell. Her pain began to fade. This is not science fiction. It is science, carefully unfolding, slow, cautious, real. It's not perfect, not yet, but in every trial, in every test, there is a breath of hope where there used to be none. Imagine a child born with inherited blindness. What if CRISPR could enter their eye and gently rewrite the error before light ever fades? Imagine a body prone to tumors because of a broken gene passed down like an unwanted gift. What if CRISPR could mend that gene before a single cell turned rogue these are not simple fixes. DNA is layered. Genes interact. Changing one line might shift the whole paragraph. That's why scientists proceed with care. Each therapy must be tested, not just in cells, but in time, in how long the changes last, in whether they ripple. But the early signs are promising. Doesn't bring healing with a shout. It works in silence, in the marrow of bones, in the fluid of eyes, in the tiniest crevices of life, and its impact, if it holds, will not just be felt by patients. It will reshape medicine itself, because if you can fix one gene, you can fix many. You can imagine a day when rare diseases are not rare, when inherited suffering is optional, not inevitable. And yet there are limits. CRISPR must be guided. It cannot guess. It does not cure everything. And not all illnesses come from DNA. Some come from trauma, from lifestyle, from chance, from the world. But where the code is broken, and where the match is known, CRISPR can be the tool to heal it. Is the molecular version of hope. Small, sharp, focused, able to reach places medicine never could. Already, trials are underway for muscular dystrophy, for rare blood disorders, for certain forms of inherited deafness. The results are early, but they whisper a future where a diagnosis is not the end, but the beginning of something better. But there is more to healing than tools. There is timing, there is choice, there is access. Even as CRISPR advances, it must become available to all, not just the wealthy, not just the lucky, because suffering knows no class, no country, no race, and if healing is to be real, it must be shared. This is the challenge that follows innovation, to bring it from the lab into the lives of everyone who needs it. But still, the path is opening, and as it opens, it invites deeper questions. If we can edit disease, should we also edit risk? Should we change genes not only to cure, but to enhance? To make muscles stronger, hearts more enduring, brains more efficient? At what point does healing become designing? And who decides? These questions live not in the lab, but in the mirror. In the quiet place where science meets soul, they lead us toward the next bend in the story, where ethics begin to glow like soft lanterns in the mist. But for tonight, let your thoughts settle gently on this idea that illness may one day be met not with fear, but with a whisper, a small correction, a kind rewrite, and that your body's story, long written in silence, may still be edited toward something kinder. In the stillness of power, there is always a question. Now that you can edit the code of life, should you? Science is not just a series of facts, it is a mirror, and every breakthrough reflects not just possibility, but choice. CRISPR is no longer a distant dream. It is real. It can enter a cell and rewrite its message. It can correct errors, restore function, relieve pain. But what else can it do? It can change the color of an eye, the strength of a muscle, the sharpness of memory, the traits not tied to illness, but to preference. And with that realization comes a stillness, a pause not of awe, but of weight. This is not fear. It is responsibility, because every tool, no matter how delicate, can be used in ways its makers never intended. What happens when the line between therapy and enhancement blurs? Is it healing to fix a gene linked to low intelligence? Is it ethical to select embryos with better traits, not for survival, but for success? And who defines what is better? What is normal? What is worth keeping? What is worth erasing? Science can offer edits, but it cannot answer these questions. That part is up to us, Already some countries have begun to draw lines, laws that forbid editing germline cells, those that pass genetic changes to future generations, because those edits are permanent. They do not stop with one child. They echo forward into children not yet born, into choices not yet made. It is like dropping ink into a river and knowing it will color every stream downstream. In 2018, the world watched in quiet shock as a scientist in China edited human embryos to resist HIV and let them grow into babies. It was meant to be protective, but the method was unapproved, the safety unproven, the ethics unclear. The scientific community recoiled, not because the dream was wrong, but because the path was rushed. CRISPR can offer beauty, but only when guided by care. This is where ethics must step in, not to halt progress, but to hold it steady, to ask, are we protecting life, or shaping it for our own image? Are we choosing for healing, or for control? These are not questions for scientists alone. They are for parents, teachers, lawmakers, and you, because the future is not built in labs alone. It is built in living rooms, in policy rooms, in quiet moments of reflection. Imagine two futures. In one, CRISPR is used wisely. Diseases shrink, suffering fades. Children are born with fewer burdens, but not fewer rights. Each edit is weighed like gold. Each choice is slow, transparent, shared. In the other, CRISPR becomes a commodity. Traits become trends. Genes are bought and sold. And humanity, once diverse by nature, becomes curated. The question is not which future is possible, it is which one we choose. To answer, we must hold CRISPR like a candle, close enough to see, but carefully enough not to burn. Ethics is not a cage, it is a compass. It reminds us that ability is not the same as permission, that the power to change must always be paired with the wisdom to wait. Even in nature, edits come slowly, through generations, through failure and survival. CRISPR moves faster, too fast for silence, so we must speak. We must ask. Who gets access? Who decides the edits? What happens to those who cannot afford them? Will genetic enhancement widen the gaps between the privileged and the poor? And what will happen to our definition of being human if we begin to sculpt it? The word designer baby once belonged to fiction. Now it knocks, gently, at the edge of reality. And yet, even in this uncertainty, there is space for hope, for conversations led by inclusion, for decisions built on humility, for a science that listens before it acts. Ethics does not mean fear. It means forward with care, because in the center of CRISPR, beneath the molecules and guides and cuts, lies something deeper, a question of identity, of what it means to let nature unfold, and what it means to intervene, of when helping becomes harming, of when fixing becomes rewriting. These are not easy thoughts, but they are necessary, because to touch the code of life is to hold not just a molecule, but a mirror. And when you look into that mirror, what do you see? A healer, a dreamer, a designer, a steward. CRISPR does not judge, it follows instructions. But we are the ones who write those instructions. And so the next chapter of science will not be written by chemistry, but by conscience. As you lie here, drifting between knowing and dreaming, let your mind return to the gentle pulse of your cells, perfect in their imperfection, alive in their silence. And remember, just because you can change something doesn't always mean you should. But where should we say yes? Where does editing serve, not desire, but need? The answer may lie not in the body, but in the fields, where life grows wild, and hunger waits for a better harvest. The field is quiet at dawn. Mist curls above the earth like breath from a sleeping world. Rows of green rise to meet the morning, leaf by patient leaf. Roots stretch underground, silently testing the soil. And somewhere within each plant, instructions. You've seen these instructions before, the twisting spirals of DNA, the same letters that live inside your own body. A, T, C, G, nature's language, repeated endlessly. But here, the code tells a different story. Not of hearts or lungs, but of how tall a stem should grow, how sweet a fruit should ripen, how deep a root should reach for water. For thousands of years, humans have worked beside this code without ever reading it. We saved the seeds of the best-tasting apples, the most golden grains, the crops that stood tallest in the storm. This was selection, slow, patient, a conversation across generations. But now, with CRISPR in hand, we can listen more closely, and when needed, speak. Unlike older forms of genetic modification, CRISPR doesn't insert foreign genes from other species or kingdoms. It doesn't build new forms from scratch. It edits what is already there. It finds the sentences in a plant's DNA that nearly work and nudges them toward completion. A gene that almost resists drought can be tuned, ever so slightly, until the plant grows strong in desert soil. A trait for faster ripening can be slowed, so tomatoes stay firm on the vine, sweetening just a little longer. A crop vulnerable to fungal blight can be given a silent defense, not from an outside invader, but from the knowledge it already carries, dormant and waiting. This is farming with a whisper, not a shout. You're not forcing change. You're listening to the code and helping it finish its thought. In 2020, scientists used CRISPR to create a rice plant with increased yields and greater resistance to heat. No chemicals, no new species, just a tiny edit in one line of DNA. The result? More food, less loss. A harvest that could feed more mouths in a warming world. And CRISPR does not stop at rice, wheat, corn, soybeans, potatoes. All have been gently edited to resist disease, to grow in salty soils, to thrive with less water. Even bananas, fragile to extinction, have begun to receive small edits to help them stand strong against the fungi that once threatened their future. These edits are quick, but the wisdom behind them is slow, because farming is not just science, it is trust. It is rhythm. It is understanding the balance between growth and restraint. CRISPR allows scientists to act with precision, to make a single, intentional change without touching the rest of the code. This matters because plants are systems. Change one trait too broadly, and you risk altering the rest. With CRISPR, the edit is clean, targeted, gentle dot, and the results are already growing in greenhouses, in test fields, in quiet rows beneath unfamiliar skies. Imagine a world where food is grown with less water, on land once thought to dry, where harvests survive storms that once meant hunger, where fewer pesticides are needed because the plants defend themselves. This is not a miracle, it is a possibility, and it arrives not with noise, but with a quiet precision rooted in knowledge. Still, the field raises ethical questions to Dot. Should all farmers have access to these seeds, or only those who can afford them? Should CRISPR-edited crops be labeled differently? Should they be patented? Will small farms be protected, or outpaced by those with more powerful tools? These questions echo like the wind across open soil, because, even in fields, editing life carries weight. But unlike older forms of modification, CRISPR edits do not leave behind foreign DNA. They do not mix species. They make changes that could have happened through natural mutation, just faster, more predictably, more kindly. This subtlety makes all the difference. It means some CRISPR-edited crops may be welcomed where GMOs were not. It means the conversation can begin again, fresh, balanced, rooted in transparency. And perhaps it should, because the future will need more food. Not just more calories, but more resilience. Crops that can survive warming climates, unpredictable. Rainfall, soil that shifts and shrinks. CRISPR may be one tool among many, but it is a powerful one, because it works with what's already there. It doesn't reinvent. It refines. And so, under the morning sun, the plants keep growing. Their leaves still stretch. Their roots still twist. But within their cells, the instructions now carry one new word, placed softly, precisely so that tomorrow's harvest can feed today's dreams. And yet, food is only one kind of inheritance. There are other stories passed down, not through soil, but through blood. Stories of illness, of loss, of fear, whispered from parent to child. And now, CRISPR stands before those stories too, asking, can we erase this pain before it begins? There are stories that live in the blood, not told in words, but passed in silence from parent to child. From chromosome to chromosome, a mother watches her son learn to walk, knowing her father never did. A father holds his daughter close, praying she won't carry what he does. These stories have names: Huntington's disease, Tay-Sachs, Duchenne muscular dystrophy, Fragile X. They do not whisper. They arrive with certainty. They are written into the code before the first heartbeat, before the first breath. They are inheritance, not of culture or language, but of biology. And for generations, families have watched as the same shadow falls again and again. But now, science stands at a threshold, a quiet, trembling place where the next chapter might not arrive, where the disease that has always returned might not. This is the dream of germline editing, to change the DNA not of the person, but of the embryo, before birth, before pain, before memory, so that the disease is not delayed, but erased, not managed, but removed. It begins with a cell, a fertilized egg, just a few hours old, a cluster of potential, no bigger than a grain of salt. Within that cell, the entire future waits: hair color, height, the arc of a smile, and sometimes a fault, a mutation that means illness, or a life cut short, or a body that won't work the way it should. What if, before that cell divides, you could edit the gene, fix the single wrong letter, prevent the entire arc of suffering? What if a child could be born free from the shadow their family has always carried, not a treatment, a prevention, a full stop before the story even starts? This is not science fiction. The tools exist. CRISPR can make the edit. Cas9 can find the faulty gene. The repair can be made. But it is not simple, and it is not without weight, because changing the germline means changing every cell in that future person, their heart, their skin, their voice, their children. It means the edit is passed on, a permanent inheritance of human intention. This is a kind of power humanity has never had before, and so we ask, what if we get it wrong? What if the edit causes harm that unfolds not in one life, but in generations? What if the child grows with a silent flaw no one could foresee? What if one pain is prevented, but another is introduced? And beyond safety, what of meaning? Does removing a disease also remove a story? Some argue that disability is not always suffering, that to erase a trait is to erase an identity, that what makes us human is not perfection, but difference. Others say that choice is a mercy, that if you could spare your child what you endured, you would. Both are true. Both deserve space, and so we tread carefully. Because editing an embryo means editing a life that cannot yet speak. It is a decision made on behalf of someone else, and yet in some corners of the world it has already begun. Researchers have used CRISPR to attempt germline edits in embryos that were never implanted. The science was real. The implications, staggering. Imagine a child who will never know what they were spared. Imagine their children, and their children's children, all free from a disease that once haunted their bloodline. It is a breathtaking vision, but one that must be held with a trembling hand. Because legacy is not only biological, it is ethical, it is cultural, it is deeply human. And yet there are places where the pain is so great, the cost of doing nothing feels heavier than the risk. A couple who has lost 3 children to the same disease and cannot bear to try again. A family with a 50% chance of passing on a fatal gene. In these moments, CRISPR offers more than science. It offers relief. It offers release. Still, the question persists. Can we separate hope from hubris? Where does healing end and designing begin? We return to the line we saw before, between therapy and enhancement, between protection and control. The difference may lie not in the tool, but in the intention. To remove disease is not the same as to select for beauty. To prevent suffering is not the same as to sculpt perfection. But in practice, the line can blur, and once an edit is made, there is no undo button. That is why most countries ban germline editing for reproduction, at least for now. To wait, to study, to understand, to ensure the child born of intention is not burdened by it, because legacy is more than what we pass down. It is what we choose to begin. As you drift now, think of the family tree of all the branches behind you, all the stories written in blood and breath. CRISPR offers the chance to prune one thorn before it grows. But the choice is not just scientific. It is deeply personal. It is deeply human. It asks, what do we owe the future? And what happens when we begin to write its first line? Some stories may begin with a cell, but others begin in the wild, in coral reefs, in forests, in places where CRISPR has arrived not to heal the body, but to mend the planet. Far from the white walls of laboratories, beyond the clean floors and humming machines, there is another kind of silence, deeper, older, and vast. The forest, the reef, the field, where wild grasses bow to the wind. In these places, nature writes its own code, not in journals, but in survival. For millions of years, life has adapted without permission, without pause. A moth darkens its wings to hide from soot. A tree thickens its bark against flame. A coral glows brighter as the water warms, a last shimmer before fading. But now, change comes too fast, too hot, too wide. Warming oceans bleach coral reefs white. Invasive species overrun ecosystems. Diseases sweep through bat caves, batons of extinction, and nature once so nimble struggles to keep pace. This is where CRISPR has begun to tiptoe, not with conquest, but with collaboration, to lend evolution a tool it never asked for, but may quietly accept. Imagine a coral reef, once vibrant with color, now ghost pale, heat-shocked, fading. Scientists ask, can we use CRISPR to help? Can we edit the coral's genes to make it more resilient to rising temperatures, to let it hold its color longer, to keep its algae partners close? Experiments begin in controlled tanks, fragments of coral edited ever so gently, not to change what it is, but to help it endure. If successful, these corals might one day be returned to the sea, not as strangers, but as strengthened kin. This is not the only place CRISPR has stepped in. In Hawaii, a bird called the ikiki faces extinction from avian malaria, carried by mosquitoes that were never part of its ancient world. Could CRISPR help here too? Not by editing the bird, but the mosquito scientists are exploring gene drives, a method where an edited gene is passed down more aggressively until it spreads through an entire population. Imagine a mosquito that can no longer carry malaria, or one that cannot reproduce. A few edited individuals released into the wild, their edited traits moving through the swarm generation by generation. The goal, to end the disease, to protect the vulnerable, to give the bird and the child in a malaria-prone village a better chance. But this, too, carries weight, because the wild is not a laboratory. Ecosystems are entangled. A change to one thread tugs at them all. What if removing mosquitoes affects the food chain? What if the gene spreads beyond where it was meant to go? Nature does not draw borders, and CRISPR, once released, does not return. That is why every wild application must be approached with gentleness, with respect, because the line between helping and harming is thin as a strand of DNA. Even so, the hope remains to protect bats from white-nose fungus, to save chestnut trees from blight, to bring back traits lost in captive breeding, the sharp eyes, the strong wings. Some dream even bigger. Could CRISPR bring back what has already gone? The passenger pigeon, the woolly mammoth, the dodo.d-extinction, they call it. But others ask if we cannot protect what still breathes. Should we reach for what no longer does? Perhaps the wiser path is not resurrection, but restoration. To use CRISPR not as a time machine, but as a bridge between what we have and what we may still lose. To make plants more resilient to drought, to help fungi break down plastic, to give trees the tools to thrive in shifting climates. These are not acts of domination. They are quiet collaborations. You, listening now, are part of this world too. The air you breathe was exhaled by leaves. The food you eat was grown in soil stirred by worms. The forests hold your oxygen, the reefs your balance. When CRISPR enters these places, it enters your story too, not as a cure, but as a conversation, one that must be spoken slowly, because when we edit the wild, we are not just shaping life, we are shaping living systems, and those systems remember, they ripple, they respond. So every CRISPR project in the wild must ask, what is the goal? Is its survival, balance, profit, who benefits, and who might be harmed? Can the change be reversed, and should it? These questions echo like birdsong in morning fog, not loud, but persistent. They are the sound of science becoming stewardship, because the world is not ours to control, but ours to protect. And if CRISPR can help do that, then perhaps it belongs here, not as master, but as guest. Still, the line must be walked carefully, with humility, because once released, These edits write themselves into the story of life. And some stories, once begun, cannot be unwritten, so we test, we measure, we wait, not out of fear, but out of reverence. For the reef, the field, the breath of a species on the brink. And as night folds gently around you, know this: the wild is not separate from you. Its survival is your own. And if we learn to edit with care, to whisper, not shout, then perhaps we can help nature keep writing its own story. But even as we look outward, a quieter revolution continues within, inside your own cells, where the future waits, soft and unwritten. You are quiet now, still beneath the covers, breathing gently in the hush of night. But within you, the future moves. It stirs, not in leaps, but in tiny threads, in the winding helix of your DNA. In the silent memory of your cells, you carry a blueprint older than fire, a pattern passed from ancestor to ancestor, never paused, never broken. 3 billion letters, each one folded into the next, carrying the secrets of your blood, your breath, your becoming. And now, for the first time in history, that code can be rewritten, not by fate, not by accident, but by design. This is not the future of science fiction, no glowing screens or superhumans. This is the quiet revolution, where disease begins to falter before it begins, where your cells remember differently, where their instructions are healed without leaving a scar. CRISPR is not loud. It does not arrive with thunder. It arrives like breath, soft, invisible, essential. You may never feel it working. You may never know what it changed. Only that one day you did not get sick. The mutation that once meant certainty is now a line rewritten, and life continues, uninterrupted. This is the future that lives inside you now. Imagine a child born with an edit so subtle it leaves no trace, only absence. Absence of pain, of struggle, of the long shadows cast by history. Imagine a body that repairs its own faults as they form, a system that corrects itself with no pills, no surgeries, only instruction, not because we conquered nature, but because we learned to speak its language in this future. Medicine is not something you take, it is something you are. Your cells are the treatment, your body the cure, and the changes are inherited, passed down like lullabies, soft edits that protect the generations to come, a chain of health unbroken. This is not utopia. It is effort, it is intention. It is the slow bending of science toward empathy, because CRISPR does not cure everything. It cannot prevent every sorrow, but it may soften the edge of many. It may change how we age, how we prepare, how we plan for life's uncertainties, and not just in health. As the technology matures, we may see CRISPR in unexpected places in your immune system, edited to fight cancer with more precision than ever before, in your skin, resistant to UV damage, in your neurons, protected from degeneration. Each possibility begins with a question: what if? And then, with care, should we? These questions will shape the years ahead, not just in labs, but in dinner tables, in courtrooms, in schools. The ethics we pondered earlier will remain. They will evolve. They will guide, because the closer we come to control, the more deeply we must listen to communities, to cultures, to each other. This future cannot belong to one country, one company, one kind of person. It must be a shared future, built with open hands. CRISPR has already begun to show us what is possible. Now it is up to us to decide what is right. And as you drift now towards sleep, your body continues its ancient work, repairing, remembering, renewing. Inside you, cells divide, DNA unzips and zips again, proteins fold, energy flows. It has always been this way, but soon it may be better, not louder, not faster, just kinder. That is the promise of this quiet tool. That is the future inside. You, not a future carved in stone, but one written in soft letters. Living letters. Letters that can still be changed. And perhaps that is the most hopeful truth of all. That your story, no matter how old, is not finished. That in the space between breath and cell, between science and soul, there is still room to revise. So sleep now. Let your body rest. Let your code carry you forward. Tomorrow waits not with answers, but with possibility, and every possibility begins with 4 letters, A, T, C, and G, and the quiet knowledge that even the smallest edit can change everything.